This is Joey Heath, and in this video we're going to discuss my research project, the development of a cost-effective CRISPR-Cas9 transfection protocol for use in Danio rerio, or zebrafish. To begin, we're going to look at some introductory material to understand the background behind this experiment. CRISPR-Cas9 is, in its natural form, a bacterial immune system that is going to essentially allow the bacterial cell to remember the DNA of viral invaders and produce different materials that, along with this protein Cas9, is going to be able to recognize and destroy the viral DNA to protect it from infection. And CRISPR, the abbreviation, stands for Clustered Regularly Interspaced Short Palindromic Repeats. In a lab setting, we're able to take advantage of this naturally occurring bacterial immunity by producing guide RNAs that are able to interact with the Cas9 protein and break DNA in a way that we can take advantage of. The basis of our transfection procedure is this protocol provided by researchers at North Carolina State University. The first gene that we were attempting to edit in this experiment was the IGF-1 gene, which produces the protein insulin-like growth factor 1, also abbreviated IGF-1. This is a protein developmental growth factor that's going to stimulate cell division. It's found in most animals, and it's expressed later on in development, even beyond birth or hatching. And it's important in postnatal and adult growth as well. It's also synthesized as a pre-pro protein, which means that it's going to require some additional processing before it becomes its own mature protein. The particular component of IGF-1 that we were attempting to edit is its E domain. The E domain is a component of the pre-pro protein, as you can see in this image here. There's a signal peptide, IGF-1 itself, and an E peptide in the pre-pro protein. And after the signal peptide is cleaved off, you have pro-IGF-1, which contains mature IGF and the E peptide. In order to form the mature IGF protein, the E peptide is cleaved off. Now, the E domain is hypothesized to influence cardiovascular development. And in this study here, it's been found that a knockout of the E domain protein was found to halt heart development in mice. The second gene that we were attempting to edit in this experiment is the SLC45A2 gene, which encodes for a solute carrier linked to oculocutaneous albinism. What this particularly does is it's going to result in the melanosome's inability to produce melanin, which is going to be the pigment that causes the black striping that can be seen in zebrafish. So you can see here in this figure, the melanosomes are going to be what are actually producing melanin and releasing it from the melanocytes. But unlike this figure, melanocytes are called melanophores in fish. In 2014, a study was published that used CRISPR-Cas9 to produce partial albino mutants in zebrafish. Unlike the protocol that we're attempting to create here, the CRISPR components were delivered using microinjection. As mentioned in the last slide and in the title of the experiment, we're attempting to develop a new protocol for CRISPR-Cas9 component delivery using transfection. Transfection involves encasing the CRISPR components in a lipid-based molecule, particularly lipofectamine. Lipofectamine and transfection have been documented with CRISPR-Cas9 use, particularly in zebrafish cell culture, though not embryos. Lipofectamine has been used to deliver viruses in zebrafish embryos, though. Transfection is essentially encasing our CRISPR components in a liposome, a lipid-based molecule. That molecule gets taken up by the cell. The vesicle that's taken up the liposome and the liposome itself degrade, allowing for our Cas9 protein to get translated and then complex with our guide RNA and move into the nucleus where it will hopefully edit the genome. Moving on to our methods and results, we're going to begin with the components that we had to prepare for our CRISPR-Cas9 genome editing. These are our guide RNA and our Cas9 RNA, as indicated in the figure here. In order to prepare our IGF-1 E-domain guide RNA, we found the necessary sequences using tools such as ZFin and Nucleotide Blast. We took that sequence and imported it into Benchling. Benchling is a tool that allowed us to design our guide RNA and examine different factors about it, and essentially gave each different guide RNA a score, allowing us to pick the best one. After we used Benchling to design our guide RNA, we double-checked it with Georgia Tech's COSMID CRISPR search to make sure that it was going to be effective and not have any off-target effects, which would mean that the CRISPR-Cas9 system was editing in places where it shouldn't be. In addition to designing our own guide RNA for the IGF-1E domain, we also used a published guide RNA sequence, particularly from the albinism paper mentioned in the introduction. To actually begin the process of making our guide RNA molecules, we started by ordering the DNA sequences that corresponded to the guide RNAs that we either designed or took from publication in the previous step. We then assembled our oligos, which essentially means that we were taking two small pieces of DNA, one that corresponded to the guide RNA that would allow the molecule to interact with the desired gene, and one that corresponded to integration with the Cas9 protein, allowing it to actually interact with the protein and essentially form a fully formed unit within the guide RNA itself. This oligo was then transcribed using in vitro transcription to produce RNA, and that RNA was purified, leaving us with guide RNA that's ready to be used for transfection. To produce Cas9 RNA, we ordered the Cas9 plasmid from Adgene. We linearized the Cas9 plasmid and then purified it. We used in vitro transcription and RNA purification on that Cas9 plasmid, and that gave us our Cas9 that was ready for transfection. On the morning of fish spawning, RNA and lipofectamine mixtures were created. The first of these mixes contained Cas9 RNA, guide RNA, and lipofectamine. Theoretically, these were the groups that would produce our mutant organisms. In order to prove that Cas9 RNA and guide RNA don't act independently of one another, lipofectamine mixtures were created for each of them, and fish were treated with that mixture. Finally, no RNA was included in a lipofectamine mixture to prove that lipofectamine was not lethal to the organisms being treated with it. It was important during this phase of the experiment that we acted relatively quickly because we wanted to make sure that we delivered our genome editing materials as close to the one-cell embryo stage as possible so that we could, in theory, pass that change on, that genome edit on, to any resulting cells that come from that one-cell embryo. So we collected our embryo, and you can see the embryos here. That's where the embryo is in this example. We then decorionated all of them using an enzyme called pronase. You can see there's a shell around this embryo. That's the yolk right there. There's the embryo. The shell is the chorion. And by using pronase, we decorionated, meaning we completely remove this chorion, or we poke holes in it, which will allow access for our genome editing materials. We then divided these decorionated embryos into 4 groups. After being divided into 4 groups and placed into 4 of the 6 wells in a 6-well plate, each group of embryos was treated with one of our 4 lipofectamine RNA mixtures, as described in the previous slide. After being treated with the appropriate lipofectamine mix, the fish were allowed to age until they were roughly 6 weeks old. They were periodically captured using a dissection microscope, and any fish that didn't survive were removed. While images were captured of the E-domain treatment groups, the SLC45A2 treatment groups are presented here as they could potentially be linked to the more visual of the 2 mutations. The images that you see here were captured 3.5 days post-fertilization, and of most interest is the Cas9 RNA and guide RNA group, as they could be our potential mutants. The pigmentation that we're discussing here is the little black dots that you can see that I'm pointing out here. But it can be noted that there does appear to be a reduction in this group when compared to that of the Cas9 RNA group, the control group, and the guide RNA, especially along the head, the anterior region, and along the tail. The same treatment groups captured 26 days post-fertilization yield similar results. You can see when compared to the control, the Cas9 RNA, and the guide RNA group, there does appear to be less pigmentation lining the tail and the head region of both of the organisms in the Cas9 RNA and guide RNA group, and this is a sample of the whole entire population of fish in each of the treatment groups. Presented here is the long-term fish survivability of each of the 3 spawning trials that we did. Figures 11A and 11B represent 2 different trials of spawning the E-domain mutant groups, and figure 11C represents the albino or SLC45A2 treatment groups. As you can see with A and B, when organisms were treated with both the Cas9 RNA and E-domain guide RNA, there was 0% survivability. while there was some survivability for both the Cas9, guide RNA, and control groups in both of these. Unlike what can be seen in A and B, Figure 11c shows us that organisms treated with Cas9 RNA and SLC45A2 guide RNA, there was roughly 25% survivability, which matched the Cas9 RNA, though there was relatively low survivability for the guide RNA and control groups. One of the most important parts of this protocol is the mutant confirmation process to confirm that there was a successful mutation made based on your transfection of the guide RNA and Cas9 RNA. To do this, a fin clip was taken at roughly 6 weeks of age from the caudal fin of these zebrafish. A DNA extraction was performed, and PCR was used to amplify the target region of the DNA that was around the mutation site. Also designed around the potential site of mutation is a restriction site. When we imported our sequence into BenchLink and made our guide RNA, it also provided information on restriction sites that could be made near the area of mutation. While we were unfortunately able to complete this part of the project as a result of the truncated semester, the next few slides detail what we should have seen had we been able to complete this project. The DNA sequence here has a restriction site in it pictured in pink. If we take that very same sequence, induce a CRISPR-Cas9 double-stranded break, and allow that sequence to undergo non-homologous end joining, we could potentially see some base degradation. It's extremely likely that we would see base degradation, which would essentially remove the restriction site, making the restriction enzyme non-effective. So if we were to take the wild type here with the restriction site and the mutant, which has had, based on non-homologous end joining and base degradation, the restriction site removed, and we run these two samples on a gel after being treated with the restriction enzyme, we would potentially see the following. Well 1, which is the wild type, would be split into two different bands because we're essentially cutting that PCR product in two. Well 2, containing the mutant, would be one band because the restriction enzyme is no longer able to function and cut that DNA sequence into two. Moving on to the discussion about the data gathered during this investigation. Starting out with some important observations, I wanted to highlight that there is likely a strong importance of the E domain in the presence of different organisms. So we can see that after 60 days, there was 0 organisms surviving in what we believe to be E domain mutant groups, and all other organisms that were spawned and treated with everything but the combination of Cas9 and guide RNA in both of these different separate trials were able to survive. It's also telling that IGF-1 and the E domain concurrently are present later on in development, so that explains why we were able to see some survivability of these different fish a few days into the developmental process, but as they began to get older and older, these began to die off relatively quickly. Another important observation made during this study is that we believe we've generated partial albino mutants with the organisms seen in figures 9 and 10 here, and when compared to the other organisms as seen in the slide that had those data in it, there does appear to be a reduction in pigmentation. This would, however, need further confirmation through the mutant screening method that I described earlier. The final large-scale observation that was made during the course of this experimentation was the validity of transfection as a method of delivering CRISPR-Cas9 components to zebrafish embryos. The hypothesized reduced pigmentation seen in the SLC45A2 mutants, as well as the 0% survivability of the potential E-domain mutants, likely lead to the fact that something is occurring, most likely the change that we've predicted based on our guide RNA design and delivery, based on this transfection process. Moving on to some of the next steps and continuation of this project. First is the development of a successful mutant confirmation screening method. While we weren't in control of the situation that led to us not being able to complete this work this semester, it's absolutely critical to the viability of this protocol that this is established as soon as possible. Also, something that can be done after confirming if an organism is a mutant or not is to do gene expression analysis. Initially, we were intending on examining whether the E-domain mutation that we were trying to make influenced the expression of various developmental growth factors relating to vascular development. This could be used for that specific mutation or any other mutation that someone would like to make. Also, it could be of value to transfect with higher concentrations of RNA, both Cas9 and guide RNA. Our concentrations are relatively low, and it might increase the efficiency of our CRISPR component delivery if we increase the RNA concentrations. Another thing that could be done is to test different lipofectamine products. We use Thermo Fisher's Messenger Max reagent, but there are several different types of lipofectamine products that can actually do different things, so that could be adapted really easily into this protocol to test those different products. As mentioned in the introduction in the last slide, we really don't know what the E-domain's proper function is. We just know that it has something to do with vascular development, so that is a site of further investigation to look into exactly what the E-domain has to do with vascular development. Finally, an off-target analysis screening system could be developed so that we can test the guide RNAs that are made and make sure that the guide RNA and Cas9 system isn't editing the genome at a place where it shouldn't. I'd like to thank the A.J. Palumbo Student Endowment Fund for providing funding for this work, the St. Vincent College Biology Department for providing additional funding, equipment, and endless moral support. Particularly, I wanted to thank Mrs. Molly Grace and Miss Kayla Uvegas. Anytime that I needed something, they immediately would help me, and it really helped the project more than it can seem. I also want to thank Grace Leonard, the world's best research assistant. Without her, this project would be absolutely nothing, and I can't thank her enough for her help. And finally, Brother Albert Gahr, thank you so much for your patience, wisdom, skill, and everything that you've taught me, not just throughout the course of this project, but also over the last 4 years. If you have any questions, please forward them to me or Brother Albert, and thank you so much for watching. I hope you enjoyed.